I did my modeling project on the synthesis and regulation of thyroid hormone. The thyroid gland plays an important role in regulating the body's metabolism and calcium balance. It curves across the anterior surface of the trachea, just inferior to the thyroid cartilage, which forms most of the anterior surface of the larynx. The thyroid gland produces two hormones, triiodothyronine, referred to as T3, and tetraiodothyronine, or thyroxine, also known as T4. The thyroid produces more of T4, around 90%, while T3 is 4 times more potent. Follicular cells transform amino acids into thyroglobulin proteins that help the thyroid gland function. The follicular cell groups can also transform iodine and amino acids into thyroid hormones that travel through the blood to help the body regulate the metabolism and excrete waste. The follicular cells surround a follicle cavity that holds a viscous colloid, a fluid containing large quantities of dissolved proteins. A network of capillaries surrounds each follicle, delivering nutrients and regulatory hormones to the glandular cells and accepting their secretory products and metabolic waste. Follicle cells synthesize a globular protein called thyroglobulin and secrete it into the colloid of the thyroid follicles. Thyroglobulin molecules contain the amino acid tyrosine, the building blocks of thyroid hormone. The first step in thyroid hormone synthesis is the trapping of iodine molecules from the diet. Iodide is absorbed from the digestive tract and travels through the bloodstream to thyroid follicular cells. TSH, sensitive carrier proteins in the basement membrane of the follicle cells, actively transport iodide ions into the cytoplasm. The iodide ions diffuse to the apical surface of each follicle cell, where they are converted to an activated form of iodide by the enzyme thyroid peroxidase. This reaction also attaches 1 or 2 iodide ions to the tyrosine portion of a thyroglobulin molecule within the follicle cavity. Tyrosine molecules with attached iodine ions become linked by covalent bonds, forming molecules of thyroid hormones that remain incorporated into thyroglobulin. The hormones T3 and T4 both contain iodide to be carried out to the rest of the body. Follicle cells remove thyroglobulin from the follicle by endocytosis. Lysosomal enzymes break down the thyroglobulin and the amino acids and thyroid hormones into the cytoplasm. Amino acids are then recycled and used to synthesize more thyroglobulin. The released molecules of T3 and T4 diffuse across the basement membrane and enter the bloodstream. About 75% of the T4 molecules and 70% of the T3 molecules entering the bloodstream become attached to transport proteins called thyroid binding globulin. Most of the rest of the T4 and T3 in the circulation is attached to albumin, or transthyretin, one of the plasmin proteins. In negative feedback, the hypothalamus releases TRH, also known as thyroid-releasing hormone, onto the pituitary thyrotrope cells in the anterior pituitary gland. The cells then release TSH, also known as thyroid-stimulating hormone, into the bloodstream. It then diffuses out of the bloodstream into the thyroid gland. The thyroid gland is then induced to release T4 and some T3. T4 enters the bloodstream, causing the body to reach homeostasis. Once homeostasis has been reached, T4 then feeds back into the pituitary and the hypothalamus and reduces the release of TRH and TSH.